Oh. So I think we'll just have to push on and hand over to Cris.
Yes. Yes.
Okay.
Sure.
Right. Thank you.
Let's see. Sure. And slideshow. It's correct, no? Yes. Yes. Perfect.
Yeah.
So thank you so much, Susan, for making available the server. Will be the first time that I will be running Reads2Map, so I'm very excited, but a little bit scared too. I hope everything goes fine. And I would do like just a presentation just to understand where it's coming from, like why we created this. It's a collection of workflows that it's made for making everybody's life easier, just for doing the simple calling and genotype calling. And I started this work when I was in my PhD. So I came from University of São Paulo in Brazil. So it's and also I also work close together with Marcelo Molinari and Gabriel in NC State. So it's actually a result from this big collaboration between us in Texas A&M and everybody. So I have a lot of co-authors. And here's my mail. So if you have questions, if you have issues, now or later, you can send me questions and suggestions. So we start this work because I started to build linkage maps with GPS data at the beginning of my master's, and was deployed species, what's easy, but was not that easy with sequencing data. So I was using at the time this package called 1Map, and it's a like old software. It comes since 2007, and was created in the lab that I come from in University of São Paulo, Brazil. And I put this picture because it's an old picture, so Marcelo is very young.
It still looks like that.
Yeah, so this tree was the first ones to create this package. And this package is able to deal with inbred lines and also highly heterozygous populations, like outcrossing, and able also to deal with biallelic and multiallelic markers. But at the time, so these are their names. At the time, we used like few markers created in wet lab. So to adapt this software to GPS, I had some issues, like using like this huge amount of markers that comes from sequencing. I had like this specifically issues, like it was very computational intensive because the package was not prepared for dealing with millions of markers. And time consuming, yes, because we use hiding Markov model, it can take a long time. And also, I would having like wrong grouping, wrong ordering, and inflated linkage maps. And so this was a challenge. I needed to use this package to build my linkage map, and I was complaining a lot. And then I learned it like, okay, be careful what you complain, because then I became the maintainer and started to be responsible for doing the updates in OneMap. And once I started to be the maintainer, I started to receive like these questions and suggestions from all people, because OneMap is very user worldwide. And I started to communicate with everyone, and everyone working with different species was very cool, because they sent me their names. Sometimes I didn't know what it was, and then I did to Google, and it was funny. But they are all having the same issues, like people starting to use like GBS or sequencing markers and having these issues with OneMap, like mostly inflated maps. So, and then it was very clear for us that these inflated maps are coming from like genotyping errors. And also, Marcelo keep working with expanding the OneMap functionalities to map to polyploid species. So he created another package called MapPoly, and he also had issues, that the same issues that I was facing, like too much genotyping errors in sequencing markers. And this new package is much more complex, and it's able to build linkage maps with a polyploid species. Jiken will talk more about it. Marcelo and Gabriel, it's the ones that are doing updates in this package. And and I was doing the updates in OneMap, and Marcelo and Gabriel was doing the updates in MapPoly to try to deal with these issues that we were facing, but and we did many things inside the package. So I tried to summarize all the tips and tricks to deal with sequence data in the poster that I presented in the Rosaceae conference. And if you want to access, it's this QR code. This presentation will also be available, so you can see like the overall picture of tips and tricks. And the beauty of working with linkage map is that we can clearly see if something's wrong. And we can clearly see because we know meiosis, we know how genetic works. And we have these pretty graphics that show a lot about how if the process is being correctly made or not. So these are a recombination fraction matrix. And this middle one is the perfect one. It's beautiful. Like here you can see like markers in the x-axis and also the same markers in the y-axis. And the cells are the recombination fraction between them. So you would expect that neighbors markers have lower recombination fraction because they are closer located. So we expect this pattern for a good order, a map that has a good order. So it will be in the diagonal will be red and gradually will become blue. That's increasing the recombination fraction between them. But what happens is that sometimes we feel we see incongruence between the genomic order and the order made by recombination fraction. So you can see in this other graph here in the left that you have like a break in this pattern here, so something wrong. But something like this, you can just understand that's the inversion, because if you come and you flip this region, you can have the order as supposed to be again. So this we can explain biologically, like, okay, my population has a genome that's different of the reference genome that was used to assembly. And you can fix that and you see, oh, it's an inversion here, and you can like take notes and like discuss this in your discussion. But sometimes you see something like this in the right, like this I can explain, like it's not inversion, it's not translocation, it's just bad data. And we use that and this specific graphic that show us a lot to identify the genotyping errors. And so what we did was using this as benchmarks, but the thing is, like, if we did all that we could in the softwares to try to fix it, to try to obtain the best recombination fraction, and even like that, we see things like that, it means that, yes, if you only have bad data, you will have bad maps. Like, it's input and output, and there's no way of just the package, just our package fix that. And it was a bad news because many things can be wrong in a GPS data, in sequence data. Like since while you make your libraries and you put your adapters, the PCR can also be very biased. And also we have all the steps for the bioinformatics steps that we know, like the quality control, the alignment, the SNP calling, and the genotype calling are also known as dosage calling. And in this part here is not under my power, but I can do something about the bioinformatics, trying to find the best way or best practices for avoiding genotyping errors in this part here. And that's what Reads2Map was created, like to try to find the best practices for this part so we can have good data for starting doing the linkage map. So, and then we do like these two ways. We use the map to see if we are doing this right, and then we correct the process, and then we do the maps again, and then, oh, now it's better, and then in changing. But I needed like automatic and like production scale way to do that. And then we created a Reads2Map. So I will explain a little bit of each one of the steps before we go to the hands-on thing. So for the SNP calling, what we do, we get these sequences and then we realign to a reference genome. And depending on the method, you see different patterns. So this would be a profile of whole genome sequence, and they overlap to each other. And this is profiles of exome sequence that you will only sequence the exomes, but we overlaps just part of the sequence. And this other one is the GBS. That's what we are talking about. And what happens that will be overlapping sequence, but only in the enzyme cut site. And they will very close to each other. It's fine. Echo? No, okay. So they will be considered duplicates because they start and they end in the same position. And this is important information because this it will need to change that compared to the other process. So in the other ones, you can see that they don't start and end in the same position. So they are not duplicates. And they usually remove the duplicates from the process. And for GBS data, we can do that. So it's something that we will see later. And after doing the SNP calling, like finding, like by the alignment, you find the polymorphisms, you need to call, like having your genotypes of your samples for each site. And for diploid data, it will be kind of easy because, or you have homozygous or homozygous classes, or if you have both alleles, you have the heterozygous. But for polyploids, you can have more classes of heterozygous. You have different doses. So not only the homozygous classes, but you can have dosage of 1, dosage of 2, and dosage of 3. And higher your ploided, more heterozygous classes you have. And you need a way to differentiate these different heterozygous classes. And what we do this type of plots. So we have like reference counts, allele counts in the X, Y, the X-axis, and allele, and the alternative in the Y. And you can measure the proportion. So if all counts, it's the reference allele would be homozygous. If half and half, it's the it's half alternative and half reference, it would be heterozygous, and so on. And then if you have a tetraploid, we will have 5 possible axes, and we will distinguish according with the proportion. Okay, and this is the hexaploid. You can see that higher diploider, more complicated it becomes. And this is a theoretical graphic because what we see in the reality is something like this. And it's a mess. And we need software to try to figure out which classes is which. Okay, this is real data I took from roses and potato data. And you can see also that in this graphic, higher the depth, like higher the number of counts that you have, it's easier to distinguish the doses. Okay, so higher the depth, theoretically would be easier to distinguish. Okay. Yes. So I can look to them. Yeah, good idea. Okay, and then okay, so we need to do that. We need to know the doses and the markers and try to figure out where they were. And the technical difficulties on that is that there are many software to do all of these steps. There are many software for doing the quality control of the sequence. There are many software to do the alignment, the SNP calling, the dosage calling, and also softwares to do the fine manipulation, like because each part has a format and you need to convert, you need to get index. So it's a lot. And all of them, so I'm talking about sequence data. So it's usually big, like it's usually terabytes or like it's big files. So it can become very complicated if you want to do like figure out the best practice because there are many. And then I needed to figure out ways of making it simple. So for the large files, I would just use higher performance computing or cloud systems. And so that's why like Susan and Ben and Cecilia made a really nice job to make a server available, because I would not be able to run in a personal computer because the files are big. And for now, like for this practice, the files will be small, because even like that, I don't want to like make you wait 1 week for finish this analysis. And also I don't want like to overload the server. And for this many software and programming language and different operational system, what I made was using containers. So containers is a very good, it was my brother that taught me to learn that, because what's a container like very in short, it's instead of I just giving to you a software and like you figure out how to install it, I'm giving you the entire environment. I'm giving you the operational system, all the dependencies, and the software. So you don't need to concern about install dependencies. That's why, like, if for the server that you are using, we are only using Docker. It's the only thing that I asked for Ben to install. Because if not, he would need to install all that softwares that I showed here. And because I'm using all of this in all the steps. So the technology of containers is something that helped a lot. And other thing that this is good is that this container has freeze versions. So I control if I want to update or not. Because what happened that if you have many dependencies and you are developing a software, if some of the other developers of your dependencies decide to update, it will break all the rest. So what happens that in containers, everybody will keep the same. And of course, we need to keep this updated, but I decide when I want to do that. And we will be using Docker, but Docker needs super user permissions. And so most of the HPC that probably you have access will have Singularity, but Singularity works in the same way. Just don't need super user permissions. And if you are doing other analysis that's not included here, many of the developers today already make available containers. So I included here a repository for containers, because you can look if you want like to do whatever, you can look there if the developer did the container. So you don't need to install. Other issue is that there are many steps. So each one of the steps that I told you will be many possibilities, and the manipulations of the intermediary files can be hard. And for solving that, what I did was use a workflow system. So I can remove that and use one of this. I use the WDL that was developed from Broad Institute, but there are other possibilities. What this makes is like make all the pipeline organized. So I have the tasks, and then the inputs of the outputs of one task will be input for the other task, and so on. Very organized and communicating with the containers. Okay. I use WDL that's called as Widow, but there are other available. And I went to a conference that they were all there discussing like, no, mine is better, mine is better.
Of course.
Yeah, and then I just stuck with the one I learned. And what I heard from them is that WDL is easier for people that come from biology, so it's me. And and then I keep with that. And other thing that these workflows allow is that you can use local servers like your HPC, but you can also use cloud environments, and they have interface for that. It's prettier. I will be hard here. We'll be going to the terminal, but it's also possible to run these workflows in their web platform. You just submit the workflows and your inputs, and then you have lots of graphical, pretty things. But we will not do that. We will go hard. We will do in terminal, because I think that most of us have access to HPC instead of cloud services. And there are also for the HPC, if you ask for your IT people, there's also a server version for the Cromwell, that's the manager of the workflow, that allow you to just press buttons and submit the workflow and submit the inputs. But we will do the hard way, okay? And so other thing that these workflows allow is to distribute better the resources. So instead of like we are used to use only one computer with like 8 cores and a bit of RAM, but these analyses require much more than this. And usually they are separated in nodes. So each node will be a computer, and inside these computers we'll have lots of cores and RAM. So we need to optimize the way we are distributing our tasks and be able to use all the resources that we have. Because sometimes it's hard, like, okay, I know how to parallelize my analysis, but I don't want to submit many scripts to distribute this in the nodes, and the workflow will do this for you. So this is Reads2Map. This is what Reads2Map do. You have like your sequence data, and then this will go to the alignment with BWA. We do the SNP calling with JTK and Freebase, do the genotype calling with UpDog, PolyRed, and Supermassa, and do the linkage map with OneMap or MapPoly. And okay, so now I will present a little bit of what we got from it, like in the paper, and then we go hands-on. So with that, what I did was I run everything. Like I run many maps and with different data sets. And the result is the recombination matrix, recombination fraction matrix. And like I could choose which one's the best because I had all these graphics. So this is with GATK. This is using GATK and as SNP calling and PolyRed as dosage calling. This is the same, but with UpDog with as dosage calling. And you can see that sometimes like this graphic's not as good as this other here or this other here. And this was with deployed rows dataset, and it was a pretty nice dataset. And I ran just a subset because I just want to know which pipeline I will use. I don't want to build the map yet. But once I decide the method, I can go in R and build the map. But I was, as I told you, I was trying to find the best filtering. So I ran many maps, like it was just simulated was like 10 to almost 11,000, and for the empirical data was 800. And I was just changing the parameters, just like to lapidate everything. So I figured out some filters that could be very useful that provides me better recombination fraction matrix. And even like sometimes, even if I have a good recombination fraction matrix, I can have inflated maps, as I told before, because of the genotyping are still there. And if I don't, but I can see that I don't expect this amount of breaks in my haplotypes that I'm estimating with the linkage maps. And then other thing that I tested was changing parameters inside OneMap, inside MapPoly, because both of them work with Harding-Markov model. And it's possible to change error rate for when you are building your linkage map. You can tell the chain, no, I don't trust too much in these markers. So don't consider the information that I'm giving to you, consider more the genetics, and it will change the genotypes for us. This is a graphic that Jiken made with his populations, and he's just track how much of his genotype his genotype call is being changed by the chain. So all missing data will be imputed, but if you increase the error rate, you have more genotypings changing. That's the red dots. So what I did with the workflows was trying different error rates to see which one of them provides me the best linkage maps. And I figure out using 5%, it's usually very flexible and it provides better maps. So it reduces, as you can see, this group is supposed to have 38 centimorgans. So first I was having, like, with the default parameters of one map, I was having 540, and then I tried to use the updog probability, because updog also output probabilities, and change a little bit, but 5%, it's closer to what I expect. And then I fixed the haplotypes. Very good. And as I say, I tested many things, like these people creativity is infinite. I just need, okay, to publish the paper, say, stop to have ideas, I will not change anything else. But we also test, like, what happened if we keep segregation distortion. We also tested what happened if we forget some contaminant samples there. I also test what happened if we have multi-allelic markers. And in this case here, I was trying to show that sometimes the inflated maps will always be wrong because we don't expect too much breaks. But sometimes small maps are also wrong because we can underestimate breaks and overestimate in other places. So I just did a bunch of simulations to show that we need to actually correct the genotypes before. And we cannot, like, tell the chain, fix everything for me, like increase the error rate, increase a lot the error rate, because the chain will do what it thinks is correct, and sometimes it's not the correct. So it's important to give good data to the linkage map. We publish, we still didn't publish, it's preprint, but it's under review, but it's available. And if you want to know more about all that we figure out, it's there. And we run with different data sets. And for different data sets, we will have different results. So I cannot tell you, like, okay, run GATK, that's the best for you. Or run freebase plus updog, that's the best. I cannot tell. Like, each data set is different. So, and this is still diploids. This is Aspen data set. And this is for the tetraploid roses. So what... that's why we have Reads2Map, because you can run your own data set and figure out your best practices. And this is the link for the tutorial. So if you want to access this, what you will see in Reads2Map, I will open here. This is where it's home. And I always, when I do updates, I do the releases. So it's basically this that you need to access. The files for running, it's all here. And come back here. So the workflows make all the pipeline be modular. So we have the tasks, that's the smaller unit, and a combination of tasks will be a sub-workflow. And a combination of sub-workflow will be pipelines. And I store these in different directories. You can run it separately. So imagine, like, I don't want to run all this functionality. I just want to run GATK. And we will find a sub-workflow for GATK here. Or I don't want to run, I want to run just freebase or just updog, or just, you will find different levels of pipeline scripts. And here's just an example of one of them. So we will have the input file, and this is all the parameters that you will need to concern about. And like for this one is the SNP calling, we will do that. You will see that in the hands-on. And it's the reference genome and all the indexes, and a file saying where is your sample. And then you can choose, like, how many cores you will use, what's deployed of your species, if you remove or not the duplicates. So this is GBS data, so we will not remove the duplicates. The replace AD is a type of filter that I explain better later. If you run the GATK, if you run the freebase, the hard filters, that's for GATK markers, the number of chromosomes that you have, because I admit Fabiana here. Yes. If the number of chromosomes, because freebase parallelize the process among the chromosomes, and the size of the chunk to distribute in the nodes, And if you run mchap, but I will not give spoilers, I will let Tim talk about that. And then with a single command, you can run the entire workflow. You just provide the input file and which workflow you want to use. Okay. This is just Freebase example. Here is the directory of tasks, and I'm showing how a task is structured. So it has like these components, the input components. I would just tell which one of the, what are the inputs of this task. And just below it, I will save the amount of disk and amount of memory that I will use according to the size of the inputs. So you don't need to concern about thinking of how many cores or how many RAM in the HPC that you will work, because I am doing this according with your inputs. For Freebase, this doesn't work very well. I can tell you, because it's not possible to predict. But with JTK, it works better. And then we have the command by itself. That would be the one that you run in the bash scripts. And it's usually, and that's a good thing too, because if you don't want to run the workflow, you can copy what I did or what I think is the best, just looking to this part of the code. Like this would be the thing that you run in your bash script. So if you don't want to use all the work, you can at least see what we think was the best. The runtime will connect with the Docker image, because each one of the tasks will be run inside a container. So the Docker, it's described here. And I also control the number of CPUs that will be used, the memory, the disks, and this will be different according with the management system. If it's learned, will be in a way. If it's cloud system, will be different. So, but I included for all of it. The meta will just describe what the workflow do, what the tasks do, and the output will be the files that I want to track. And to run a single task, you can use minwdl, and it will you just put the input files that it's this session and the task file, and then it will run for you. And you can get the output file, that's the VCF. And you can combine, so this is one task. You can combine the task connecting inputs with outputs and create a workflow. So this is the Freebase sub workflow. And the task that we just saw is this run Freebase. And as you can see, like I am connecting each one of the tasks and creating a sub workflow. And here, this first task, I am distributing among the chromosomes, like I'm splitting the band files in batches by chromosome, because each chromosome will be run in a different node, just to optimize. And then I run the Freebase, and then I merge the VCFs of the chromosomes, and then after I do a normalization with VCF tools. And then the output is the VCF file. And just the same, here is the workflow. So I put it not only the Freebase sub workflow that I just show you, but I include the GATK too. So I'm just like including more things. And you can run separately, as I can show here. And that's it. We'll start to do with your own hands now. And I want to thank all the co-authors, and they come from different places. And I also want to thank Susan, Cecilia, and Ben for making the server available. Have other project members and collaborators. And here are the other tutorials. If you want to do SNP calling, genotype calling with other softwares that's still not included, there are other tutorials that you can see. And also the workflows, it's very nice. I like it. And there are other tools that you can use for management and for tracking what it's doing. And there are other links here. And my brother did one very nice that's for tracking if it's working, if it's not, which part of the workflow is running in the moment. And it's nice. Okay. So I don't know, the link is here. And we should open this. I try to make this tutorial very self-explained, so if you want to see in the future, you can. So I included some Linux commands that we will be using in the description, all of it. So if you have questions, if you don't know what it means, you can come back here and check. Not only for Linux, but also for Docker, because we'll be using Docker. Here's the link for the requirements that you receive in your mail that Susan sent to you. If you want to use to install this, everything that's required, it's described here. Okay. And then you probably have access to the server. Yes. Everybody's following? Yes. We will use a terminal. So just open the terminal.
Here's his refresh. You want to refresh it.
Yes. So you should see something like this. Good. So we will first create a directory so we can organize it only in this one. So you can copy here and paste here, and you will have a directory. And the directory, I will need to download the examples. So wget will download from the link. So you just come here, copy and paste. Now we have 4 passkey files and a FASTA file. So the FASTA is my reference genome. It's all subset, so that's why it was so fast. But oh
no.
Oh.
Just put it up. Yeah. Next one. Okay. It's just kind of team. It's just yeah, you can just. So, if you do get a issue like that where it says that it's timed out, you can dismiss rather than restart, because often it just loses the connection for a little bit, and then you'll still have all your
Okay.
Sorry.
Oh. So things say that when it happens with server, just press dismiss instead of restart, because then you don't lose your. So here's an example of what thing was just press dismiss.
Yeah. And see.
And now you want to get those files, so just copy and paste all of those things into that. No, yeah, yeah.
I won't worry about it. I want to go to the
The document. Okay, so links to the chat routines.
Should it be my computer?
Yeah.
Just
No,
to make it
it's
flow?
sorry.
Chat, so
Should it be my computer?
Oh, you can run on the computer.
Can you, it'd be easier to not go through, can you do it in terminal? Can you SSH to their terminal? Maybe.
I can do my computer, so just to keep the code, but others do.
This is great.
Yeah.
No, she doesn't actually have the files on there, but we can send you information. I have the whole.
Okay, so that's
Yeah, but she's actually pulling the FAST files, and so that makes the FAST files. You can just manually type them. All right, so you go to your links. I think, yeah, it's putting the best way. And is thinking maybe it's the internet.
That's weird. So whenever we did in each other's
so I have you from your own terminal.
maybe 10 minutes, and we're on the same floor. Come on.
That means
I have my.
Probably because I sent links.
Needs some heavy checking.
I think I'll do that, doing my own terminal, but it's the same thing. So I'm changing here. I will do it in my terminal. Okay. File, and then just create
File. File new. And then you want to terminal.
Okay.
Yeah. Okay, now we want to make a directory, so we just make folders.
Oh, it's
And then you can, yeah, okay, so we want to just repeat them. So this way.
oh, okay.
Comment here.
So you have just
I didn't know.
now we've made the folders. Now we want to go into the folders that actually CV. Yeah, can you close?
Maybe can you?
Okay, just
And now we want to get the files. So we need
just search Reads2Map.
So you easy. You can just copy and paste this all up. And so you want to be in a new
Just go to her page and we'll find it.
be in a terminal.
I think I also, it's stuck.
So you say RGC in the repository.
You want to be in exhibits.
Yeah, RGC.
Use the bash instead of the
These files.
Is
So don't forget, it's just how we do this around.
Yeah.
where is it on your? Where's the thing on your GitHub?
What?
Where's the HTML? She can't. The HTML.
Okay, now you can see.
And then we should open the slash. So I think it's gonna.
And the bar. And the, not the pipe, the one that's angled.
That's okay, now you can try again.
Someone in my user.
And Reads2Map 2023, and then you should access.
Should be 7.
No, you're in user 35, there you are.
Do you have it?
I have the full link. I tried to email to it, but it's not going.
Okay, you have the bar, like just put the bar.
Yeah. That is why I
Type in
can't do it.
Reads2Map 2023. So I think the reads, the R is, yes.
This should be.
We go back.
Did it work? Maybe? No, let me pick it. Drop it to you.
Oh, yeah.
I don't know about there. There, I got it.
Just on
Did it work?
There you go.
Got it, got it.
Mark.
And then let me airdrop you the link to it.
Nice. That's pretty.
You can just repeat. You need to run, run. So you can essentially copy that whole thing. Okay. And then press run.
Just press enter.
And then
Maybe it works.
Yeah. Awesome. Thank you.
I ran that thing as I could. But
Bad student.
Yeah, no, I'm a bad student. No, well, I didn't have time. Download all the articles, everything else. I was learning the concepts. I'm watching. I'm watching. All right, you can help us. He's got hope.
Can we keep going?
I'm exhausted.
oh,
Good, great.
Can we keep going? Everybody's okay?
it doesn't work.
People online, if you have questions, just send in the chat. Susan is looking, other people else is looking. If someone doesn't have the link, it's this link here for the tutorial.
Okay. Yeah.
Now you can do what you need.
Yeah.
Based on when all done, right?
So the first step will be just checking how the sequence are, because we want to know if this sequence work, like if you have good quality and so on. So a good software for doing that, and it's very famous, is FastKC, and it will provide us reports for each one of our files. So I did a loop here, so I will run only one command, but it will go interactively by each one of the files. I just wanted to show you before the formats of what you expect. What was the sequence files like? It can be abstract, so we can just check the FASTKey files. It's composed by this. Each sequence has 4 lines. The first line is the header that will inform the position, the name, and so on. And the sequence by itself will start in the second line. That's the ATCG that everybody knows. And the other line, like this FF or these strange letters, is just coding for the quality. It depends of the platform that you use, but FastKC can understand every platform. And that's it. And then start another sequence. So each sequence has 4 lines. And you have a full file with a bunch of sequences. Okay? This is FastKC format. FastKey format. The genome will be in FASTA format. So this is only one chromosome. And by sequence of the FASTA file, we start with this plus or minus signal. And this is just chromosome 4 of roses. And I have just a piece of it. But if you have a genome, you have more plus signals and the name of the sequence and the description of the sequence. Okay? So we will just, of course, we don't want to see each one of the lines and everything. So FastKC will do a report for us. We'll summarize the quality and what it's composing your sequences. Okay, so you come here. In the server, they installed the Docker, so we are using the Docker. Okay, but I put the option for if you have Singularity in this tutorial, because I don't know if online people have access to other HPC, because Singularity is more common to find. Okay, but I just put here to you to know how to use in Singularity.
So just
Yep.
So now the last.
And I don't know how it works. I'm using this Docker image, the biocontainers FastKC. And if you don't have in your user, probably what will happen is that you will download this first. And you don't need to do anything. It will do for you, but will take more time.
So now what should?
I'll just go and check what happens. Yeah, you already. Perfect. Can you go a little bit? Okay. Yeah, you didn't need it to download. Good, we'll be faster. So we are not there yet. So it's going up.
It is a way to the class to sequence to the class cases.
Yeah.
Yeah. This. So now next thing.
I'm great at hosting. I know. I'm gonna have thanks to good tutors.
Okay, everybody, okay. Nice. So all of it. So, and you will have like, if you go, oh, sorry, I'm not doing the anymore, but you have the HTML files. And you can open in the server, you can go and open one of them just to check how is the report.
Do you want to show them how on in Jupyter Lab notebook?
I have one here.
Oh yeah. So did you show them how to do it in the Jupyter Lab notebook tab? If you click on here, yeah, it should be in there. It should be in there on yours.
Yes. Yes. Here.
So in the folder, yeah, like you press that, and then also in there. Yeah.
I sit on.
You're in the HTML is the fast to see
And
it.
then you press it.
This is the.
I think she'll explain what they are.
I have one video.
Click twice.
Yes.
And yes.
First time for slide one.
Yes, like I just reviewed it. So it was there's enough you go through that so you can actually the base.
I would just 10 minutes. 10 minutes good?
Yeah, whenever's good.
Whenever it's good. No, fine.
Okay, so the report, you have like these items. Here you can see the quality of your sequence. So the line is the mean of your sequence. They do like this overall. So you can see that the sequence have a good quality because all the sequences is in the green area. And we have like the other ways of seeing the quality. So all good. This, I will not explain each one of them because we want to keep going. But what I wanted to show you is that sometimes you have like error report, like saying that's bad, but it's actually just because it's GBS. So if we have like whole genome sequence, we would expect that each position of the sequence is random, like if you're having C, G, or T, it's random. And this graphic showing that it's random in the other bases, but at the beginning, it's a huge bias. But this is because we have the cut site. So every sequence will have the same base there. And they report to saying that's wrong, but it's just because the method that you use it. And it's the same thing for duplication level. It's saying that's wrong, but it's because in GBS, we use the PCR duplicates. So it's here, so you don't need to concern about that. The duplicates, we also change the GC peaks. So it's a mess. And for whole genome sequence, you would expect like this smooth curve, or if you have contaminants, you'll be like two curves, but do not expect something like that. But this happens because of the duplicates. So it's not thing that you should concern. Okay, if you want to be sure, of course, you can do other analysis to see if there is no contaminants. But this is not a good graphic to detect contaminants just because it's GBS. And you have like this issue with overrepresented sequence, of course, because we have a lot of PCR duplicates. So, but anyway, it's a good report for you to check the quality, check if you have adapters, because sometimes you still have adapters. So it depends on what you see here. It's what you would do next to trim your sequence, to make sure that you are providing the alignment and SNP calling good quality sequences. I included in the tutorial some suggestions on how to do, like if you find like adapters, what you do. If your sequence doesn't have good quality, what you do. And that's it. I'll just get my tutorial again here. I will just, instead of doing the break now, I will just run the tutorial, the workflow, because it takes a while. So when we go to the break, we just let it running. Okay. So to run the workflow, what we will do, it's load the files. So the files that we are loading is this one in the GitHub repository. So I always do a release. And then we have here the WDL, the main pipeline. All the tasks, all the sub-workflows that the main pipeline use will be compressed into zip files. So it's the dependencies. And I have a template for the impute file also. So if you run this command here, you will download all these files. Okay. You come here. Oops. And you should have here now the empirical SNP calling imputes, the WDL, the zip file, and also this sample info. So this sample info is just telling me where the files, the FASTQ files are, which are the name of the samples. And if you have like a duplicate, like sometimes people run same sample in different plates, you can define which one is unique and the name of the sample and which plate was, because GATK consider different plates when calling. And here's the input file. You can, this is a template. I included here the parameters that we use for running this workflow now in the server. But you can change this with parameters that you want. It's just important to know how much resources you have available and how you want to distribute your analysis according to the nodes and the CPUs. Okay. And for running, first we need to create the genome file index. And I will need lots of other extensions. This file is just support for the genome. And I will use 3 different images for doing that. So I will run this. Yep. So if you see here now, I have not only the chromosome 4, but chromosome 4 sub FASTA, but I have also the extensions of DICT. And all other, that I will not pronounce all of this because, but it's all auxiliary files for the genome that will be required for doing the alignment and the SNP calling. And then once you have it, other thing that we need to change is the back-end configuration of the workflow. So you have a file here that calls, you should have this Cromwell, this one here. It's the default here. This is where I configure all on how the workflow is communicating with your system. If you're using Singularity, you should include the default as this Singularity and Learn. And I also have the local and configurations for SGE, that's other management system. You can also have Docker and Learn, because sometimes HPC has Docker instead of Singularity. I don't have all the possibilities here, but if you need, if you have a system that uses another management system, let me know that I can include here configurations for that. Right now in this server, what we are using is the local. It's the first one because it's just only Docker. There's no management system, so we should change the default to local. And for doing that, you can open the file. This file is the Cromwell no MySQL, because yes, we can connect workflows with a database if you want to keep cache, but we are not doing this right now. So the only thing that you will do is to change here to local. Okay? And save it. To save, I press Ctrl S, but I don't know how to save without the shortcut.
We're doing that now, or we would do that in the future?
No.
Right now.
No, do it now. Okay, I just came here and opened the file and changed it here.
And if you save this now, it's just our version saved.
Change to local, right?
Yeah.
You just want to change this first line. Yeah.
Local.
To local. Save it.
Just do that.
Control is to save it.
Yeah, Control S. Whatever
Control S, that's the
Yeah, so
This one.
when you save something, so when you get unsaved, that X up there, compose the file will be a dot to show that it's unsaved. So once you save it,
Yeah, so Control S.
Good, I reopened it.
Yeah, and see, so when you're changing, there is a round circle, but when you save, then it's just a cross.
Means you save.
Yeah.
So what she was doing to look at the files. So
Oh, just the old.
Good, very good. Change to local.
looking into the file inside your terminal, so you just listen and but you can't modify it.
Yeah.
Okay, once you did that, we have the WDL. We choose the WDL that's first need calling. We have the view file, and just to show you, we are choosing to run, yes, the Freebase. We are running also the GATK. It's true here. We are filtering the markers with the hard filters. I have a link here that show what's this hard filtering, and you can check this later. Because I followed the best practices of the GATK, and these documents show which were my filters, thresholds, and how I did it. Okay, the link is there. And so once we have all these files prepared for what we wanted to run, we just run this command. And actually, I will run this one because Ben made a variable, and because I'm using the Cromwell. Cromwell will manage all these workflows. Cromwell will make it happen. I usually just use the file, but we have like this variable, so just run this second command here.
Second.
Yeah.
This one.
Although we have to first do this, we have to index that files. So do this one.
And then this will print lots of things in the screen, because we'll tell you in each part which part of the workflow it's running in the moment. But you just let it run and go grab a copy, okay? Oh, hello everyone. Hello. Are you back? We are here already. And we checked the computers here around the room, and some of them failed, some of them no. So if you don't have, here, this is my screen. This is the end of the workflow. And if you have a failed in the last line, means that yours was one of the ones that failed. And if you don't fail, you should have your outputs here. Okay. And if you are the case that failed, it happens because I think we overload the server. We didn't have enough RAM memory to run everything. So some of the process ran and some of them failed. And if a single task fail in the workflow, you have all the intermediary files for the other tasks, but the workflow you say that you failed. So probably one of the GATK parts, that's the haplotype color, it's more random consumer. And if probably what happens that we didn't have enough memory. So some of you have results, some of you don't. But I would not say to run again, because the next part, we will not use the outputs from this part. We could, but what happens that I did subsets. The first part, I subset the individuals. So you just have 4 individuals, 2 of them are parents and 2 of them are populations. We are working with F1 autocrossing population, but I didn't want to run like 200 samples because as you can see, we already fail with only 4. So for this, I subset the samples. And now we will do the dosage and the linkage map. And I couldn't subset the population because if I do that, we would not have any results. But instead, I subset the chromosome, which just be a single part of chromosome. And that's why you need to download a VCF file. But theoretically, what we would do in real situations that we get these VCF files that's output from these workflows, and we keep going with the other workflow, okay? But in this case, it's not connected because of the subsetting. And the workflow, like, do you remember that the tasks, I say which are the outputs here. So each task will have output. And in this case, it's VCF files. And the ones that I'm including as output will be, the path for this file will be described here. And it's a little scary because yes, the path is long, but it needs to be long because I'm doing many things at the same time. So the workflows organize the directories in a way that databases or interfaces can access each one of the results. So this path here is, I output the plots for the hard filtering that you can see here in this documentation that I showed you before, that how GATK describe how they filter. So if you come here in this path here, and here you will see all the figures for showing how your markers are being filtered according to the hard filters. You can do this, or you can do in the hard way and come here and just check. You see that it's NIP calling, the ID for the workflow, and then is GATK genotyping.
Sorry, I just have to interrupt you a little bit because of the memory time. I don't know if it's too late to take this 12 gig memory limit. That's in the workflow kind of quick. Processes are trying to access. It's good, no matter what. Max I don't know if it's worth doing that or not.
I think,
Are you good now? Have you gone past that step?
I don't know if people want to make it work because I think that running the workflow again will take time. So even if it fixes.
Okay, yeah, so you have to start again. Yeah, no, okay.
But thanks.
Right, just in case.
Yeah, he's great. Ben's great. It would.
Yeah, it's just some processes being killed, but then you just have to try it again.
Mm-hmm.
Yeah.
Yeah. Okay. Thanks. Thank you.
Yeah.
Okay. Ben figured out a way to fix all the fails, but I think we will just keep going because if not, we'll take too much time, and I want other people to talk to. Okay. So if you want to access the figures, you just follow the paths, and these plots will be like in the hard filtering. Here, call plots, execution, call plots, and you can access the graphics for the quality of how was the filtering made. And of course, all the tasks has outputs, and I tracked just the main outputs. And here in these lines, you see like the quality plots, the merged band, and something that MCHAP use that you can take this merged band to input in MCHAP later. The VCF files for Freebase and also metrics for the VCFs. for Freebase and for GTK. And here are the main VCF files for both of them. So you could just get the files from here. And I would just open one of them to show how is the format of a VCF file. So this is a VCF file from JTK. The VCF has a header, and the header will describe each one of the metrics that it's containing in the VCF files. So you have description for each one of the measurements, the values. And it depends on the software that do the SNP calling. This can change a lot. And the information by itself starts after the reader. And then you have like chromosome information, position information, ID, what's the reference, what's the alternative. And after this first information, I think it's 9 columns that will give you information about each marker. It starts the genotype by individual. So as I told you, we have 4, in this case, I think 3 individuals. And then here will be the genotypes. This is missing data, but we have like a genotype like this one, that's dosage 1. And we have other metrics that we can see what is each one according to the description of the header. Okay? And I think probably Tim will talk more about PCFs later.
Okay, so you just need to put CD in.
Okay?
And then, so this if you go like that.
Questions?
Where are the results? I can't.
So the output, like at the end of the log file, you have this part here
Trying to access your user.
that describe the path for the main outputs. Like all the intermediary files will be in the Cromwell execution directory that's here located. Oh, sorry. Come back. So we are running in this tools 2023, and the workflows create automatically these 2 directories, Cromwell execution and Cromwell workflow logs. The logs is the same thing that you are seeing here in the screen. And the workflows execution will have all the intermediary files that the workflow outputs, including the main outputs. The main outputs, I decided which ones it is. And I decided that VCFs were important. So the workflow will print the path to the main workflows, main outputs here. So if you get this path here, you will find your VCF files.
Yeah, and then not
Okay.
the end of the line, just the.
Merge VCFs.
Okay, so this is VCF file. So the last one is normalization.
Yeah, that's what.
Text file.
Steps.
And I think
Yeah.
we go into. Just go into
And then biological
yes.
normalization.
Yeah. Copy that.
And the execution.
And then, yeah. And then it's that.
That's
Okay.
It's all right.
Yeah, it's easier when you
Now
get
we're
the path in the log file because it gives you all the directions. And here and there. Okay? Good? Yes.
I'm just trying to get into that picture.
Have you found it?
Yeah.
You found it.
I think so.
It's normalization.
Normalize.
It's this.
Everything, right?
Yes.
She found it.
So,
Oh, okay. Yeah, but need to be your on your log.
Yeah, that's what I'm doing.
Calling.
Can you? Do you just want to go there?
JTK.
Yeah. And then
As you can see, a simple word
Okay, now you see things. All right. So that's on VCF.
Programmer, did they go to?
Did they go too fast? Okay,
Yeah.
sorry.
Yes.
So this is our output from our JSP course. And then
After that,
Did you find
the
the files?
Yeah.
This is the files.
you only have three, I think.
Okay, let's see.
That's it. This is using that. That's what you must see. I want to see.
Great. So you don't have. You don't have content.
So she's gonna tell us next.
Important, so you miss the.
You are.
So we just read it like the thing that we did before, the outputs, we just go into the. Yeah, I usually
In
don't.
the middle?
In the middle, how? This one?
No.
And then you open that one. And then you open the typing it.
Oh, you got it.
Normalization.
Yes.
And then what's the next? Normalization.
Oh, you don't have output. You failed at the very last step. All the way down to the folder and then the output.
So one thing that's,
Other things.
yeah, it's very good, but I didn't show, but I have a tutorial for how to do that, is how to connect a MySQL database to the workflow. So if something like that happened, like the last task of your workflow fails, you already have a database. So if you run again, you don't run all the rest. You just run the one that fails. And this is very useful.
So can you specify from where? Or it will automatically
It
check?
will automatically check.
So
because
we don't need to.
Yeah, the database will understand what tests were failed and what tests were okay, and then it's just run the ones that will fail, all the ones that you change it. If you change some task and then you want to run again, it will understand also. It's like Makefile, like it will understand. Okay.
We just
Okay.
Yeah. Me too.
What time is supposed to finish?
11.
11, okay.
But you can take some. The sun's kind of out of mind.
Okay, I will just run more of these comments. What we will do now, like, okay, we had the VCF files, and now we will, these VCF files, the dosage is according to JTK in freebase. But sometimes can be a good idea to run other software that takes into account the segregation of the population to fix the dosage. And this would be the Supermassa, Polyred, and Updog. And remember, we are comparing them. So at the end, we will know which one's the best.
So yeah, so she's showing all of these.
So just need to download this other data set. And now I'm downloading the empirical maps. That's other workflow that will do all of this for us.
No, we're looking at your space and the server, so it should be the exact same. But it doesn't matter. You are in the folder, so that's true. I don't know why it's actually visible there.
Okay. And then now we have this input file.
We can see it on the menu.
That I need to inform which is deployed, which ones are the parents, because it's a mapping populations, F1 outcrossing population, which chromosome I'm mapping, and like also the number of cores that I will use, and I provide the VCF files. So this VCF file is the ones that I just downloaded that have more individuals, And I also have like other parameters, like filters approach, like I'm filtering here by genotyping probability. And if I will filter by the every time that JTK do that, if it has a missing data as the genotype, the allele counts will still be there, but will always be the reference allele. So I fix that in the workflow. If the genotype is missing, I put the counts as missing too, just to not messing up with the dosage calling. So you have many options here. I have all described in the tutorial, and I put links there so you can explore more later, okay? And to run this, we will do the same thing. So now we have the input file. We have the workflow that we want to run and all the dependencies. And our configuration file is already set, but according to what we have. And then we come here and we just run.
It's very good. I know.
And then, as you can see, it creates automatically other directory. That's the directory that will contain the results for this new workflow. In the same directory, the Chrome executions. Now you have maps and snip calling. If it happens that I run again, it will create another ID. So this huge number here, random, it's an ID for your run. And if you run again the same workflow, it creates another ID. So you can keep tracking the runs that you have it. While it's running, I can show you the tutorials. I try to keep it updated because the paper is under review. So I am.
Share the link.
Yes.
Just.
So I try to, I'm changing a lot because of the reviews, but I try to make like the vignette match what you will see in the versions. Okay. And here I describe like with these graphs what the workflow is doing, which tasks is performing, what the sub workflows have, and all the. If you want to understand deeply what you are running, because you are pressing a button, but it's doing a lot of things, as you can see in the distribution of the directories. And then if you want to understand each one of the steps, if you want to include other steps, it have a way to, like, it's like programming. You have many levels of how much you can learn. You can just run and get the VCF, but if you want to change something, if you want to, you need to understand a little bit more what's happening behind. And then I try to provide ways of doing that too.
Sorry, I'm not very good at it.
It will take some time to run, but if you have questions.
Yeah, so. And then we are now going to be doing text. So we just need to get these.
I will share some links in the
copy and paste that.
chat.
Oh, waiting to rerun.
Yeah, okay. Yeah.
Good?
Yeah.
You're good?
Yeah, it's still working. Still rerunning the previous one.
You could have another break.
Let's go back to this. And then what we want to do is.
Okay. The whole thing?
Yeah.
Yes.
So it's still waiting, so you would go back to that base or batch. So you have the $ at the end.
Oh,
Yeah.
great to get this go back to
yeah.
I have all the
So
presentations
actually,
there.
for later, I wasn't able to install the development.
That's it.
Yeah, can I have you?
Yes. I
Okay, so the
Yeah,
first
that's
thing is that
great.
we need to see. Yeah, the French also is. So the first. F2s, but we see that with this F2, so we are just as. I think you can actually share more package. I'm forgetting that in.
Because as you said at the beginning.
Decide on
Just for the.
It's like sometimes I
Why is that we all change the first waiting for return code?
Someone is.
I used to start with the left.
And then it's so different. The log is like just waiting on the.
Exactly.
And
wait
then
for
with these,
the-
I can
Yes.
paste,
Yeah.
like,
Exactly.
I can just
So because. So then.
Shut down, what's wrong? Time out.
Language that it's not as complicated.
Oh, your log is up.
Remember last? Remember that? I work on this.
And then
It's kind of like.
cannot
it's like-
EQ.
This is the descendant.
Mine's running Macaulay now.
But I cannot remember all of the commands.
Should I start, restart?
I think because it's gonna, we can't see the results. It's the log.
So folks that work in this, I mean, they're just concepts.
Yeah, yeah. You have to read.
Do I need to rerun that?
Get someone else to learn for you.
What will happen?
We are almost finished.
Right.
If mine runs, I just can show the results.
Okay, that's fine.
Yeah.
Still busy.
Yeah.
But it works.
Where should I test the result? Completed all
It's all in the execution.
the same?
Chrome execution.
The same? The same?
Yes, it will put all the results there.
I think it's still
And
thinking.
now you have maps.
Linking, just.
Like all the IDs, and even if you didn't finish, you have all the intermediate
Problem
ones.
now. I'm not gonna remember at this point, really.
Then you can add this doesn't have a last because it's branches, so you have steps for all of them. So you can to upload polymaps.
To save everything and then to look.
Lots of things that we can. This is the questions that we ask.
That's what I'm doing research now.
Any help?
Execution.
Just when-
And this didn't finish. This doesn't have. Yeah, yours was break it in the middle.
Send it to the background.
Yeah. Yeah, you can like just put the plus and direct to the log.
So if I run the command again, it's just like you said, continue.
In this case, no, because we didn't connect database.
Oh, so everything that you restart.
Yeah.
Will it generate another one that looks like a shutdown or something?
Yes.
So the not,
I was
it
just
should
thinking.
break.
But we could do a database. I shouldn't, I have this in the tutorial, how to connect. We just need to install the MySQL also. That's why I didn't want to give too much work, so.
MariaDB, MySQL. Yeah.
Yeah, and then you just open this in another Docker image and connect it with the workflow, and then it's done. It would be only running the ones that fail. I usually do that because never know.
Especially when you're handling large.
Something question that I just.
Yeah, it's failing. I will reinforce to use database because the MySQL. Because if you make mistakes with the resources. Yeah. Okay.
So there's this error message that package I have version.
I'm to run.
Yeah.
So
Yeah. People online, I don't know, I'm talking in many different places of the room. I don't know if you're listening to me, but some of the tests are failing, and the reason why is just resources. And it can happen, of course, like if we make mistakes in the resources that we have, it will happen. That's why it's important to connect it to a database. In the tutorial, I describe how to do that. You can just open another Docker container with the MySQL and connect this. I just didn't now for the workshop because I didn't want to give too much work and to the people that were configuring. But this is a good thing. Like you can, and then what happens that if you have a database connected, if the workflow fails in one task, you can fix the memory that you are using and adapt this, as Ben was saying, and then you don't need to run all the other steps. The workflow will run only the tasks that failed if you have the database connected. Okay?
Yeah, just remove it just by default you don't want to do that. So do you want to?
We have the chunk. We have
Yeah,
the
sure.
chunk.
Okay, let's do it.
I think it's just taking a long time. Yeah, it's just taking a long time.
Okay, it's going.
Because
Usually by default you prefer to say no.
exact same thing.
And
Well, simple, it's good.
so hopefully.
We just wait.
Oh, you got to use that? There you go. Thanks for the encouragement. Well, it's still running. It's moving forward. What's your name?
Esther.
Maybe I can show some results because I already have some intermediary, because it's not finished mine yet, but I can show you. And at the end, we will have the list of the paths for the outputs. And then you can use these links to get the images and all the results. And in this case, I compress all the results in a tar.gz file. So it will be only one link. It's easier than the other. And it will have like images, like graphics like this one. That's graphics from Mappoly, just showing the profile of your markers, like segregation distortion, how much of the missing data, how many like are each one of the dosers, and the recombination fraction matrix. And as you can see, it's not very pretty. And I provide these graphics for each one of them. And this is for the, in this case, I think it's GATK call. And so I just took the dosage from the GATK and passed through the linkage maps after building all the metrics. And then you can just check here which one would be the best for you. And you have this type of graphic for all the combinations. That's the same graphics that I show in the slides. Okay. Good. And I think I would just let it running. I don't know. What do you think?
Yeah.
I don't know how long it could take. And I don't want to just be here waiting.
I'm very confused. How do you read that heat map? It's like.
Oh, so this is a very messy heat map.
Yeah.
So how I read that, it's not good.
I just feel, oh,
Yeah,
how do you
because-
read it?
Because we should see something like this.
Oh, so you can see the clear.
Yeah, so if it's very different of this, means like don't use this approach.
Very visual and clear.
Yeah.
Right. No.
Yeah. Because it's a subset, I cannot blame, like GTK usually gives good dosage, so don't, because it's a subset. So we will not expect very good results with this is small. But yeah, that's how we know. Okay. Okay. Can we just change? Okay, thank you. Thank you, everybody, for